So I'm very honored to be here, and thank you very much for the invitation, and very humbled to follow Jim Watson after that really interesting presentation. And I figured this was a really reasonable venue to go back in history, and in fact, back in history a lot further than we heard from Jim. And actually, just as a prelude to this, at lunch, Jim told me he wasn't really interested in anything that happened in the universe before the origin of life. But I had decided to begin with the origin of the Earth 4.5 billion years ago, and think about these events spanning 4.5 billion years. And after the molten Earth began to solidify and one of the oldest pieces of the Earth's crust was found in Western Australia, in this period here we start to get what one might call the primordial soup from which life evolved over the next billions of years. And in the first billion or so years we had prokaryotes. And some of the fossil evidence indicates that it could be as long as 2.5, 3 billion years ago. And then around 2 billion years, everything here is plus and minus 500 million years, I would say, cells evolved with a nucleus that contained DNA. And the whole point of this is that all of life that we know, and of course humans are right out here at the end, is based on DNA. So all life forms that emerged from these archaea ancestors, the last common ancestor, across all these species that exist today, and we're at the very tip of this little spike here, are based on DNA. And this beautiful molecule, for which we thank Jim and Francis Crick for the structure, as we know, adenine pairs with thymine, cytosine pairs with guanine, and at the tip of that spike with the animals are, of course, humans, and every human cell contains 6 billion base pairs of DNA. The haploid is 3 billion base pairs of DNA. That's a lot. This is about 15 base pairs. And what we know is that DNA spontaneously decays. And having that many billion base pairs in each cell, there's bound to be hydrolysis events at these glycosyl bonds that hold the base onto the sugar, of these covalent bonds that hold the amino groups onto cytosine, adenine, and guanine. And so we get spontaneous damage induced in cells every day. And in fact, in the time that it takes you to read this sentence, your cells will have accumulated 10 trillion DNA lesions throughout your body. And the assumptions here are 20,000 lesions per day per cell, 10 to the 13 cells in a human body, and about 4 seconds to read that sentence. I think you'll hear in the next talk that this estimate of 20,000 lesions per day is probably a gross underestimate. You'll hear that from Tom Kunkel. But basically, lesions are happening all the time, not just from spontaneous decay, but from exposures like sunlight, chemicals in air, food, and water that we consume, radiation, normal metabolites that exist in cells, inflammation, which happens in response to infections and other situations, and the reactive oxygen species that escape from mitochondria that are consuming oxygen to create energy. And so we're constantly exposed to DNA damaging agents in our environment, not just from the things we have to eat, drink, and breathe, but also from our ambient environment and in the clinic. Chemotherapy agents, for instance, are potent DNA damaging agents. And in the situation of an infection, one gets an inflammatory response that creates chemicals that can damage DNA. The research that we've been doing for many years has been trying to understand why it is that even if, well, we know that everyone has different exposures throughout their lifetime, But even if everyone had the same exposure, we would have different responses because, of course, we have different genomes. And it turns out that there are 6, and in fact, again, in the next talk, there'll be a 7th that you'll hear about, but 6 major DNA repair pathways that take care of all this kind of damage that's occurring spontaneously and being induced by environmental agents. And these are base excision repair, nucleotide excision repair, direct reversal, mismatch repair, which you'll hear more about, homologous recombination, which you'll hear more about, and non-homologous end joining. And it was for the realization that these DNA repair pathways are really fundamental to life that recently, as we heard earlier, the Nobel Prize in Chemistry was awarded to Tom Lindahl for his work on base excision repair, Paul Modrich for his work on mismatch repair, and Aziz Sanjar for his work on the mechanisms of nucleotide excision repair. So we have these pathways, and I will emphasize that all of these pathways, actually perhaps with the exception of non-homologous end joining, were first discovered in bacteria. These were pathways that had to evolve early during evolution in order to generate stable species. So really the ability to take care of damage that is inevitable in the DNA is fundamental to life. And it's very clear that the action of these pathways has a profound effect on human health. And the reason we know that is because there are human diseases that have defects in any one of these pathways that have dire consequences. For instance, some defects in base excision repair predispose people to colon cancer, to neurodegeneration, to skin cancer, to colon cancer, and ovarian cancer if you're lacking mismatch repair. If you lack homologous recombination, you're prone to breast cancer, ovarian cancer, and pancreatic cancer. And in the absence of proper non-homologous end joining, one can have an immune deficiency. So in order not only to sustain life is repair important, it's also important to sustain health within organisms. And also these pathways are very important in protecting against situations where we deliberately expose people to chemotherapy agents or radiotherapy for cancer treatment. Many of these pathways are very important. So from everything I've said, it's very clear that the role of these DNA repair pathways is very positive. So DNA damage, if left unrepaired, can lead to cell death and mutation and go on to cause disease, many different kinds of disease. But if repair acts appropriately and is given time to act, that can be prevented. So you don't get cell death, you don't get mutation, you don't get disease. So I'll be telling you about one more example of this from our work today, but I'm also going to tell you that under certain circumstances, the action of DNA repair can do the opposite. It can drive cell death and mutation. I'll talk mostly about cell death. And so this is a very counterintuitive and not that common, but in some circumstances, the action of DNA repair actually drives disease. So it's all about context. So we've worked primarily on alkylating agents, chemicals that transfer alkyl groups to DNA and other molecules. And as I said before, these are present in our exogenous environment and endogenous environment. So for instance, tobacco smoke, fuel combustion products, certainly some chemicals in food can alkylate biomolecules. Chemotherapeutic agents can alkylate, certain ones can alkylate DNA. The endogenous sources are S-adenosyl methionine, natural metabolites, and also in the presence of an inflammatory response where you have the release of lots of reactive oxygen and nitrogen species, one gets the stimulation of lipid peroxidation, and lipid peroxidation goes on to cause the production of alkylating agents. So I'm going to begin with inflammation, which over the past many years has become more and more clear that there's a link between inflammation and many of these common diseases, cancer, heart disease, Alzheimer's, and so on, and stroke. And if we think about inflammation, it's part of the immune response. It's something that's designed to protect us against invasion of foreign pathogens or during certain kinds of injury. One has an innate immune response, a very fast response, where the recruitment of things like cells like macrophages, eosinophils, neutrophils, these cells are recruited to the area where the infection is happening. They release lots of nasty chemicals that have collateral damage and have collateral damage in DNA. So in a situation here where, let's say one has a Helicobacter infection in the stomach, this stimulates the production of lots of cytokines, which in turn recruit and attract these macrophages and neutrophils that in their attempt to get rid of this invading pathogen releases lots of reactive oxygen and nitrogen species that go on to directly attack DNA and other cellular molecules, in particular lipids. So this lipid peroxidation that's induced by reactive oxygen and nitrogen species form these alkylating chemicals, hydroxyalkanols, that can attack DNA. So we have attack of DNA at guanine in two different positions, producing two different ethenoguanine lesions. Sometimes I call them adducts, sometimes I call them lesions. Ethenoadenine, ethenocytosine. Now, what I'm going to tell you about is some work we've done looking at the action of a particular DNA glycosylase. The DNA glycosylase, alkylating DNA glycosylase, which initiates the base excision repair pathway. So Aag glycosylase will cleave out, it was identified as cleaving out a methylated base from DNA. And I'll just quickly go through this pathway. This now produces an A basic site in DNA that's recognized by another enzyme to cleave the sugar phosphate backbone. The 3' hydroxyl here is used as a primer for DNA synthesis. There are activities to trim off these 5' flaps here. And then the final step of base excision repair is to ligate the DNA. And of course, the information on the opposite strand is used to restore proper DNA, the proper DNA sequence. So this particular glycosylase we started working on because it repairs methylation damage, but it turns out to repair, and this is just a sampling of lots of different kinds of damaged bases. So in addition to cleaving out by cleaving the glycosyl bond at a 3-methyladenine, 1-methyladenine, 3-methylguanine, 7-methylguanine, these are lesions that are pointing into both the minor and the major groove of DNA. In addition to cleaving out those bases, this enzyme can also cleave out damage that's produced in the presence of an inflammatory response where one has a lot of reactive oxygen and nitrogen species. And in particular, I'll point out these ethenolesions that are recognized by this glycosylase. Now, several years ago, Bevan Engelward, when she was a student in my lab, she's now a full professor at MIT and one of my closest colleagues, but when she was a student, her project was to knock out this repair function in mice. And she generated a mouse deficient in the Aag DNA glycosylase activity, and we went on, we and our lab and her lab have characterized this mouse for the past several years. So the thing about this mouse was that lacking the glycosylase makes it deficient for repairing damage that's induced by inflammation, and also deficient for repairing damage that's induced by methylation. And we first, well, it wasn't first, but I will first tell you about the action of this glycosylase on inflammation damage. It had long been hypothesized that the inflammation that happens in ulcerative colitis, where you have repeated bouts of inflammation in the colon and a predisposition to colon cancer, that in diseases like this, the reactive oxygen and nitrogen species were contributing to cancer because of the DNA damage they produced. And this, we directly demonstrated that this indeed is the case, in that if we take these repair-deficient animals, put them through several rounds of inflammation or colitis, we find that the repair-deficient animals have 3 times more tumors than the wild-type animals that can carry out repair. And in fact, if we just look at what happens to the tissue, you don't have to be a board-certified pathologist to see that if you can carry out repair of those atheno lesions initiated by the Aag-glycosylase, that you can, the tissue looks a little deranged, but not nearly as deranged as when the cells lack that Aag-initiated base excision repair. Now, we knew that there were a couple of more enzymes that exist in people and in mice that can act on those atheno lesions induced by inflammation. And so the ALK-BH2 and ALK-BH3 enzymes can directly reverse this damage in DNA. And so we wanted to ask, well, if we knock out, as well as taking away Aag activity, if we take away ALK-BH2 and ALK-BH3, making a triple knockout mouse, which takes a long time, would these animals be even more susceptible? It turned out these animals do not get any tumors, which was a surprise, except for the reason. And that is, they were so sensitive to even a single bout of inflammation in their colon that they all die. So they don't get tumors, but they die. And in these animals, if you restore any one of these pathways, the animals will survive the colitis and go on to get tumors. Unless they can have all 3 pathways, they get fewer tumors. So the take-home message is that the constellation of how much DNA repair you can carry out of these atheno lesions grossly changes how a tissue will respond in the situation of having an inflammatory response. So this is clearly a situation where the action of the DNA repair, the Aag-glycosylase, and in fact, the ALK-BH2 and ALK-BH3 is a beneficial thing in that it prevents cell death, the mutation, tissue derangement, and the induction of disease. So one thing I want to point out to put this in context is that if we take blood samples from healthy people in the population and in the white blood cells measure the level of Aag-glycosylase activity, you can see that the level of that activity spans quite a broad range. In other words, the natural variation in the activity of this repair pathway, at least the initiation of base excision repair by this enzyme, has a 10-fold range. Therefore, it's possible that depending how much of this you express will determine how badly or how well you might respond to an inflammatory response. So this is the beneficial aspect, and for the most part, the action of DNA repair pathways is a beneficial thing. But we were surprised to find that under conditions of alkylation rather than inflammation, the action of the DNA repair pathway actually drives tissue damage and disease. And again, we're using this same mouse, but now instead of inducing inflammation, we're inducing methylation. And the first tissue we looked at, because we were very interested in alkylation chemotherapy being particularly hard on bone marrow cells, we wanted to see how bone marrow cells responded to alkylation in the absence of this repair pathway. And we were astonished to find that the absence of repair actually made the bone marrow myeloid cells resistant to alkylation. And another way to put that is that Aag by its presence, is driving cell death in those myeloid cells during conditions that one might experience during chemotherapy. So what is our model? Why do we think this is happening? We set out, and I think we've shown pretty conclusively now, that what's happening is that in certain cell types, not all, but certain cell types, if you initiate base excision repair, but cannot get through to the last step of DNA ligation, that what's happening is you're accumulating toxic intermediates, these strand breaks in DNA that are signaling the cells to go into death. And so if you now knock out the ability to even initiate base excision repair at that lesion, the cells become resistant. And the corollary of that would be if you increase the level of the glycosylase, you would get even more intermediates and more cell death. And so we made, to test this idea, we made a transgenic mouse that expresses increased levels of the glycosylase. And I'll point out that the range of increase compared to wild type is anywhere from about 2-fold to 9-fold. That's within the range that one sees in the healthy human population. So it's not a massively overexpressing mouse. So if we look at the bone marrow, which has about 3 times more glycosylase, this hypothesis is fulfilled here. And this is what I showed you before. Wild-type bone marrow cells, somewhat sensitive, take away the base excision repair pathway. They're resistant, increase by just 3-fold above the level in the bone marrow. In the wild-type animals, you see a massive increase in sensitivity. So this is a situation where having the repair pathway is actually detrimental in the face of an environmental exposure. So we have this mouse. It has a complex phenotype. We've shown in the colon and the stomach, I didn't show you the data, that without base excision repair initiated by Aag, the animals are more sensitive, there's more cancer. In the bone marrow, in the absence of the repair pathway, the tissue is alkylation resistant. Looking at the rest of the animal, there are a number of other cell types, specific cell types within other tissues. In the cerebellum, the photoreceptor cells in the retina, beta cells in the pancreas, and cells in the thymus and spleen, all of which, in the absence of repair, are able to resist the toxic effects of being exposed to alkylating agents. So I'm going to I'm going to show you some examples of the effects in cerebellum. I won't go through all these others, but we have parallel studies showing more or less the same thing. The cerebellum at the base of the brain is, and here it is in mice, is important for providing smooth, coordinated movement. This is the structure, which is more or less the same in humans and in mice. And looking a little more closely at these lamellae here, these folds are filled with cerebellar granule neurons, which actually represent 50% of the neurons in the brain, even though they're embedded in a little tiny structure that's only about 10% the size of the brain. And it's these cerebellar granule cells that show this peculiar phenotype of being very resistant to alkylating agents if you do not initiate base excision repair. In the transgenic mouse we have about an 8-fold increase in the cerebellum for the Aag glycosylase, and these are animals all treated with the same dose of alkylating agent, MMS. If you have no ability to initiate base excision repair, it's as if the damage hasn't been administered. In wild type, you have damaged cells dying in the cerebellum, and in the transgenic mouse, you have even more death. And so one might say here that too much DNA repair activity is a really bad thing. However, by too much, we're talking about the wild type level of DNA repair, not just in the transgenic. So the, oh gosh, is there a mouse? How do I do this? A mouse. I just wanted to get a little video going on here. Oh, could you? Thank you. So the gold standard test for whether one has cerebellar damage is this accelerating Rotarod, which looks like a kind of cool experiment, but it's very tedious, to tell you the truth. And the mice are put on this rotating rod, which speeds up with time, and you time how long the mice can be coordinated and stay on the rod. The tediousness is that they have to be trained twice a day with an hour rest between the trainings, and only on the 5th day do you actually get to test their ability to stay on that rod. So with all these genotypes, the Aag with no repair, wild type, and the transgenic, in the absence of treatment, they're pretty equivalent. Exposure to alkylating agent grossly affects the transgenic animals, and if we go to a higher dose, you can even see a difference here between wild type and the Aag null animals, which can still remain on the rod for 5 minutes, as if they haven't been treated at all. So the model, coming back to this, is that the initiation of repair generates base excision repair intermediates that have this ability to signal cell death. And so we wanted to know how that signaling was happening. And one of the best candidates was to look at poly-ADP ribose polymerase. This is a nuclear enzyme. At least PARP1 is a nuclear enzyme that recognizes single-strand breaks in DNA. It binds to those single-strand breaks, and upon binding, activates its ability to generate these poly-ADP ribose polymers. With mild amounts of damage, this allows the recruitment of DNA repair enzymes that help the cell survive. And in fact, you've probably heard about using PARP inhibitors to treat cancer patients because repair is thought to be inhibited, and that makes the cells more sensitive. However, there's another flip side to activating the poly-ADP ribose polymerase, and that is when there are a lot of single-strand breaks in DNA, the generation of these polymers depletes the cell of NAD, and further, that results in depletion of ATP, and this sends the cells into either apoptosis or necrotic cell death. And so we believe, well, we showed, that in fact the activation of the PARP1 poly-ADP ribose polymerase is required for this cell death that's being generated. Here we have untreated cerebella. In the presence of the Aag glycosylase, we have the driving cells into cell death. If you take away in those animals the poly-ADP ribose polymerase, they look as if they haven't been treated at all. And so the PARP is required downstream of the initiation of base excision repair by the Aag glycosylase. So one might ask, do the inhibitors that are now in the clinic have this effect? I'm not going to show you the data because we don't have enough time, but in fact, these inhibitors that are actively being administered to patients now have the effect of diminishing cell death induced by alkylating agents in the cerebellum of these animals and in the retina as well. So our model is now that the accumulation of these toxic intermediates stimulates PARP activity, and this leads to necrotic cell death. So coming back to this model here, you can see for the inflammation, the action of the repair pathway is helping the animal, and in this case, the action of the repair pathway is detrimental to the animal. And it seemed at this point that, okay, so for the Aag-initiated base excision repair, for inflammation, it's a good thing. For alkylation, it's a bad thing. It kills cells. And so we wanted to test the generality of this and wanted to look at another form of inflammation being induced in these animals. So we chose to look at ischemia reperfusion. Ischemia reperfusion meaning blocking the blood supply to these organs for some period of time and then allowing blood flow to resume. During the ischemia, this induces an inflammatory response, but the resumption of blood flowing into tissues that have been ischemic can actually also be very damaging because you have a sudden burst of oxidative damage. And we've looked at ischemia reperfusion in liver, kidney, and in the brain. We haven't yet looked in the heart. And so this was the liver ischemia reperfusion that we set up. Cutting off blood flow to the large lobe for an hour, and then reperfusion for 6 hours or 24 hours. Now, given our results showing in the colon that the action of this repair pathway is a good thing when you're suffering an inflammatory response, we expected to see a lot more damage in the repair-deficient livers than the wild-type livers. So we assessed liver injury as anyone who's taking a statin has given a blood sample to have your liver enzymes tested, see if you have any injury to the liver. And to our surprise, the Aag-null animals had much less liver injury than the wild type. And this was true at both time points. So this was much more like the alkylation result. Now the action of initiating base excision repair by the Aag-glycosylase seems to be driving tissue damage in the liver. If we look at the area of necrosis, this plotted here, clearly there's more necrotic region in the liver in the repair proficient animals compared to the Aag-null, and another enzyme indicator of liver damage confirms that result. So, given what we'd seen for alkylation, we asked the question whether ischemia reperfusion, now it looks like in this situation we have, again, accumulation of toxic intermediates that would stimulate PARP activity leading to cell death. And indeed, if we look at PARP activity in the red here, we see more induction of PARP in the nucleus, poly-ADP ribose in the nucleus compared to the repair deficient. And in fact, if we plot this a different way, looking cell by cell, it's clear even in the repair deficient, you have some PAR polymers being made in the nucleus. This is intensity in the nucleus, but in the wild type, you have much more intense generation of these polymers. And as it turns out, you have much more intense loss of NAD and loss of ATP, which would drive the cells into energetic crisis and into necrosis. So now the model gets a little more detailed, accumulate the toxic intermediates, which we've measured in terms of abasic sites, stimulation of PARP activity leading to necrotic cell death. I'm getting there. So the thing about necrosis is it's a very untidy way for a cell to die. And don't forget, this is all being stimulated from the action of a glycosylase that's generating single-strand breaks in the DNA, and we're seeing cells dive into this necrotic pathway. Apoptosis is very neat and tidy. It doesn't result in an inflammatory response, but necrotic cell death causes in itself inflammation. And one of the reasons it causes inflammation is because some of the molecules that are spilled out in that tissue are specifically stimulating an inflammatory response. And during these studies, we saw these studies showing that HMGB1, a classic nuclear protein, turns out to, during damage in the liver, gets translocated out of the nucleus into the cytoplasm and out of the cell. And HMGB1 is this incredibly interesting protein, which I don't ever thought of it as being in the nucleus, but when it's floating around in the serum, it can act as a stimulator of the innate immune response. It actually binds to TLR4 receptors, binds to RAGE receptors, and stimulates pro-inflammatory cytokines that now recruit innate immune cells. And so if we look at HMGB1 in the wild-type animals that can do repair, you can see dissemination of HMGB1 in red. And in the repair deficient animals, that stimulation of HMGB1 being translocated just doesn't happen. And this is the quantitation here. And if you look at the recruitment of, this is HMGB1 present in the serum of animals, wild type, that have undergone ischemia reperfusion, the presence of this glycosylase throws HMGB1 out into the serum to stimulate inflammation, and in the absence of repair, that doesn't happen. And if we look at the recruitment of neutrophils into the liver, indeed, you have more recruitment in the wild type. So again, just getting a little more complicated, ischemia reperfusion causes inflammation. That makes substrates for the Aag glycosylase that generates base excision repair intermediates. Stimulation of PARP activity sends the cells into necrotic cell death, causing the release of HMGB1. HMGB1 recruits neutrophils that generate more reactive oxygen and nitrogen species and more damage, creating a vicious cycle. We've shown similar results in kidney, which I won't show you, and we've shown similar results, not as far as the HMGB1, but in the brain, which is particularly important for thinking about stroke victims. So here, the white areas are necrotic areas induced by ischemia reperfusion in the wild type, but not in the repair deficient animals. You have much less necrosis. We assume this is being generated by poly-ADP ribose activation, and in fact, it's been known for a very long time that if you give mice PARP inhibitors, you can reduce the amount of stroke damage to the brain in the presence of PARP inhibitors. And I think what we've discovered here is that the upstream actor is the generation of single-strand breaks by Aag-initiated base excision repair. So I'll just end with saying, far from a simple model where inflammation, this pathway is a good thing, and alkylation pathway is a bad thing, to we have to modify that. Certainly for colonic and stomach inflammation, this repair pathway is beneficial, but for these other situations, it's not. And the role of DNA repair and base excision repair in particular in inflammation as it's associated with all of these diseases, I think would be interesting to look at. And I would end with acknowledging these wonderful people who did all of this work: Lisiane Mera, Jennifer Calvo, Apritim Mazumda, Maria Carmela Aloka, Darini Shah, Mohammed Ibrahim Khani, Ali Danishman. This was a collaboration with MGH for the brain, ischemia, and Kimberly Fake. And I cannot give a talk at Cold Spring Harbor without acknowledging John Cairns, who really gave me my start as a researcher and was, as mentioned, former director here at Cold Spring Harbor. And my first visit in 1978 at the age of 25, nearly 26, was the beginning of me realizing that I had to spend the rest of my life doing research. And finally, the research funding. Thank you.
So thanks. The questions, we'll just have a couple of questions if there are. I think one of the things that this begins to really demonstrate very nicely that the tissue specificity of the downstream signaling from repair really matters in terms of getting cancer or one always wondered why BRCA1 mutations were specific for ovarian and
Right.
breast, but that,
And it's not
Again.
just tissue specificity. Within the tissue, it's a particular, for instance, in the retina, you have these layers of neurons, but it's only the photoreceptors that behave this way. And there's no way they're not all being exposed to the same dose. It's really remarkable.
question up to
Hi, I have a question, maybe harder to test, but if this toxic intermediate, this damage, can be suppressed by boosting up the downstream repair pathway, so yeah.
My favorite way to suppress the generation of those intermediates will be by inhibiting the Aag glycosylase. So people are currently inhibiting the PARP, the activity of PARP, but I believe inhibiting the upstream activity would be a better way to go. Then you don't generate those strand breaks in the first place. However, the NIH didn't like that idea, and unfortunately, those inhibitors are not being looked for at the moment.
So one more question, Nicola.
Yeah, maybe I can ask then. So P53 is the obvious candidate, no, as a regulator of DNA damage and so on, and it has been implicated also in inflammation, in necrosis. There is a recent report that using the same model is actually in a P53 null background, you can block it. Do you know if that actually can regulate the contextual response to DNA damage?
If we cross with a P53 null, we modestly diminish this response, but not completely. Yeah.
So my question is, given that the link between inflammation and base repair, can you find certain signatures associated with inflammation, certain mutation signatures?
I would think so. There are so many different kinds of lesions that are induced that it gets a little tricky because you have cytosine, guanine, but more or less all the bases are damaged in some way. So, but there probably is a signature. I don't know the answer to that right now.
We may hear
But we may hear about something like that in the last talk.
So in your BER knockout mice, are the immune cells normal, or do they die quickly also?
No, they appear to be normal. So the initiation of an innate immune response appears to be equal in an Aag. It's a really great question, and it's something we've struggled with and even done bone marrow transplants of the Aag null animals with a wild type bone marrow and still see similar. Yeah.
Okay, thanks very much.